All right, next up is Gary Churchill from the Jackson Laboratory. His talk is titled Post-translational Mechanisms Buffer Protein Abundance Against Transcriptional Variation.
Thank you, Clement. And I have a simpler title here, but what I'm really here to tell you today is that model organism genetics is alive and well. And now that we finally have the powerful genetic resources that we need, combined with the molecular profiling technologies that we have in hand, we can use these systems to ask fundamental biological questions on a global scale. And I'd like to tell you a little quick story how we're just getting started, and you all can contribute to this effort. Before I get into this, I really want to give great thanks to Steve Munger, who, among other things, prepared most of these slides and did the bulk of the real work behind this story. Joel Chick and Steve Geige from Harvard University, who carried out the remarkable proteomics, which you won't hear much about, except it's totally underpinning the story, and myself, I'm speaking. We all know this story. For 60 years or more, we've known the central dogma tells us that DNA makes RNA, makes protein. And if this story is true, from a genetics perspective, we should see the following kind of thing happening. If there's a change in the DNA that causes a change in the RNA, that change should then be propagated to the protein. And we should see, for example, an up mutation in the DNA that increases RNA level should increase protein level. However, it doesn't always seem to appear to be the case that when we change DNA, it affects RNA. It should affect the protein, but there seems to be some kind of disconnect. This is not a new story, but recently there's been a lot of accumulating evidence to indicate that people are starting to notice that the DNA, RNA, protein story isn't really the whole story. And of course, there are many, many layers of regulation that sit on top of the central dogma, and I'd like to tell you a little bit about how we use diversity outbred mice to get at part of this story. There's a whole talk about diversity outbred mice, but very briefly, they are an outbred cross derived from 8 inbred strains, and there's also a complementary set of inbred strains, which are recently mostly completed and available from UNC. Please see Darla Miller in the audience. There's a tremendous amount of genetic diversity, and for us the key in this population is that there should be enough genetic diversity so that there are enough changes in the DNA that we're manipulating almost every RNA. That's the key to this experiment, is that it's very global. There are so many millions of changes in the DNA that every RNA is essentially perturbed somewhere in some mouse. So we carried out the following experiment using 192 DO mice liver samples. We carried out RNA-seq analysis. We carried out proteomics. The Harvard guys carried out the awesome proteomics. We did the eQTL mapping, and then we made some comparisons. And one really striking thing, many of you have seen these eQTL maps before, where we have, I'm just going to wave, the location of the protein and the location of the QTL. There's that strong diagonal band indicating that things co-localize, the QTL and the protein coding gene. When we looked at the overlap between the pQTL and the eQTL, it was remarkably close to chance. It's almost like you just rolled the dice. This was a bit of a puzzle until we started to pick it apart into the 2 components, looking at the main diagonal and looking off the diagonal. So when we look at the local QTLs, the pQTLs almost always have a concordant eQTL in 80% of the cases, and I suspect more because we may have some power issues that cause us not to detect all the overlap that we could possibly see here. These genes typically show a strong correlation between transcript and protein. We can go back to the founder strains and validate we can predict and validate that these relationships hold, and the central dogma appears to be the story behind this large number of almost 1,400 proteins. We look at the other side of this Venn diagram, and we can see that there are several hundred pQTLs that lack local eQTLs. These tend to be uncorrelated between the transcript and the protein. but we can still predict the protein levels from the founder strains. So we can predict the protein. We know that there's a genetic signal, but somehow that genetic signal is skipping over the RNA and going straight to the protein. In some cases, this may again be a lack of power, but in some cases we validated that there's an amino acid change, or we will validate that there's an amino acid change in the protein that alters its stability. And there are probably several additional explanations for how and why this kind of regulation might occur. I also wanted to briefly mention that there are a large number of transcripts, almost 2,000, that have eQTLs but lack pQTLs. Once again, I'm going to say power is a potential explanation, but here's a pretty whopping example of the TPM3, which has a huge local eQTL, so the RNA is clearly perturbed, but the protein is not responding to that genetic signal. So there's some disconnect and some kind of buffering must be going on. No correlation. So the really cool stuff, however, is in these distant protein QTLs. We see almost twice as many distant protein QTLs as we would expect based on our prior experience with expression eQTLs. They're all over the place. There are no striking trans bands. They just seem to be scattered all over the genome. When we looked at the concordance between eQTL and pQTL in the distant, we saw quite the opposite story. There's a statistically significant lack of overlap. It's really striking. And per our last speaker and Steve's question, at least one of these we know is a translocation, and I suspect 7 out of these 9 are translocations. So these are really cis-QTLs that are just in the wrong place because we don't know where they really are except in the reference genome. So somehow the QTL here is distantly affecting the RNA, which affects the protein directly. The really interesting class of pQTL that I want to get to today is this class where we have a distant pQTL but no corresponding eQTL. These give us a handle into understanding protein regulation that is really fascinating. So somehow there's a distant genetic variant that's reaching through to the protein, skipping over the RNA, and if we dig a little bit, we can actually start to understand what are some of the steps along that long blue arrow. So basically the long blue arrow represents a regression of the target protein on the pQTL genotype, the distant pQTL genotype. And if we break that arrow apart, we can actually start to fill in some of the pieces along the pathway statistically by carrying out each of the possible regressions of all 8,000 proteins and all 21,000 transcripts that we measured. There's a bit of a logical challenge here that I'm not really going to be able to get into except to show you an example of how we sleuth out causal intermediates. So this example is TMEM68. TMEM68 is transmembrane protein 68. That's about as much as we know about it today. And it has a local eQTL and it has no local pQTL, but it does have a nice distant pQTL. So there's some disconnect happening here. Let's take a look at that distant pQTL. And the height of that peak, the LOD peak, is just a regression sums of squares. And the thing is, if we do those regressions and we start trying to fill in what's along that blue arrow, what should happen to that LOD score? If we find an intermediate, the LOD score should drop. If something's not an intermediate, the LOD score will not drop. There are lots of things that can make the LOD score drop, but what we're going to do is we're going to go through everything and we're going to rule out things one by one. So first we're going to look at all 8,000 proteins, and the height of that blue cloud up there is the same height as the LOD peak at the distant pQTL, so none of these really make it drop except one, which is a gene called NNT, which just happens to be right on the spot. So we've got a really strong prior that if there's a causal intermediate, it should be local to that pQTL, and there it is. And out of 8,000 proteins, that's the only one that could potentially be an intermediate. And when we look at the RNAs one at a time, voila, NNT also drops. And so now we have a hypothesis that can be validated in the wet lab telling us that there's genetic variation on chromosome 13 that affects NNT and cis, the RNA, which affects the protein, and the protein somehow affects TMEM68. NNT is localized to the inner mitochondrial membrane, so transmembrane protein 68 might very well sit on the inner mitochondrial membrane. It's known that this is a deletion in the B6 strain. This is great stuff I don't have time for. And there's more to do in the lab. We need to understand exactly where these steps are happening, and it's not clear whether it's happening at translation or post-translationally, but I'm going to make an argument now that it's probably post-translational, and I'm going to do that anecdotally. Here's a protein complex. It's called CCT, chaperone and containing TCP1 complex. I want you to notice the beautiful symmetry of this thing. 8 subunits, perfectly symmetrical. It's a chaperone. The CCT2 subunit has a very weak eQTL, but it has a strong distant pQTL. So you see the same story emerging. If we take that distant pQTL and we do the same mediation analysis regressions, a lot of things drop down. But they all have names like CCT, CCT, and then, oh, there's that TCP1 thing in there. I'll tell you, SUMF and ZBED5 are just due to local LD. This gives you a sense of what the LD window in a DO cross is. It's very narrow. So a whole bunch of proteins drop down, but only one RNA drop down, and only CCT6A is local, so we have a strong prior. It seems to be the case that CCT6A is regulating CCT2. Now, if we do this analysis again for CCT3, 7, 4, 5, 8, and TCP1, all the same things drop down repeatedly. And the short version of this story is that CCT6A has an allele contributed by the NOD strain, which has a very low level of expression. That low level of expression is in turn translated to its partners in the complex CCT8, 7, 3, 5, TCP1, CCT4, and 2. And this is the beautiful animation by Steve Munger to illustrate our hypothesis of stoichiometric buffering and how that might control protein abundance. So imagine that transcription and translation are making a bunch of protein subunits, but in some mice we come up a little short on CCT6As. Then we start to put these subunits together and they form their nice chaperone, beautiful, symmetric, stable complexes, and they keep doing that until at some point we've made all that we can make, and we try to make another one, and we have run out now of CCT6A. The subunit's unstable, it's going to be degraded, and not only that, but all the other proteins that couldn't get in end up in the proteasome, and poof, they're gone. That's our story. Just because I showed you 2 examples doesn't mean that's all. There are about, depending on how you count them, several thousand examples of pairs and small groups of proteins that all interrelate in some way that we can precisely define in terms of their cis and trans QTL relationships. Sometimes these small groups form extended networks. We can see the endosome complex and some of the subunits of the endosome, the exocyst complex, are all linked by these analyses. And in fact, there is a hairball that Steve's graduate student Alex Stanton drew with Cytoscape, and you can spend hours playing with it. We do. Thanks to the people in my lab, Peter, Kwangbaum, Narayanan, Dan. Thanks to Karen Svensson, who helped immensely with the mouse breeding. Alex, Steve's new graduate student. Ed Hutlin, another Harvard member of the team, and our funding from the NIGMS. And thank you.
Thanks, Gary, for a great talk. Any questions? Please come up to the mic.
Yeah, quick question. If I understand the model properly, this disconnect between RNA and protein may be more true for proteins in complexes than single proteins floating around in the cell. Is that something you see in the data?
We found a lot of the examples where there was a disconnect between protein and RNA to be interacting pairs or groups of proteins that are in physical complexes. I totally agree. That's what we see. I can't say it's the only thing that happens.
There's no microdata
Actually, the Gagi lab made one of the most extensive protein interaction maps that we are aware of, and they overlap spot on. I couldn't tell you the numbers right now. It's somewhere buried in the methods of our paper. Thanks.
Thanks, Gary.